So again, we will be discussing about plant development, and our subtopics for today will be about the meristems and the flowering plants. Okay? So, so in plants, most of the adult structures are actually derived from two regions. Okay? So the first one would be your embryonic shoot, and then the second one would be from or coming from your root meristems. So both of which are actually maintained after germination, and let's start with discussing about the embryonic shoot. Okay? So the embryonic shoot becomes the shoot of the apical meristem of the growing plant that give rise to all the stems, the leaves, and the flowers. So in flowering shoots, the vegetative meristem becomes converted to floral meristems that make flowers and not leaves. Okay, so basically when you say floral meristems, from the name itself, it would give rise to the flower of a plant. Okay, so in such, like in Arabidopsis, the shoot apical meristem changes from vegetative meristem to an inflorescence meristem. So usually yung pinaka-flower talaga, or yung kasama yung petals, the sepals, and all of the structures that you see on a flower, you call it an inflorescence. Okay? So the first stages of the future organs are known as primordia. So in singular form, it's primordium, where each consists of small number of what we call your founder cells. So these founder cells actually produce new structures by cell division and cell enlargement accompanied by cell differentiation. So if you remember from our previous topic, all of the cells or the stem cells in the plants, um, hindi talaga sila katulad ng meiosis with animals na um, same lang yung laki nila all throughout the division process. For plants, the stem cells do actually, or the growth of the plant is actually dependent on the enlargement of the cell and its differentiation. So it's the enlargement and the elongation process of the cells. Okay? So, um, So there is usually a time delay between the initiation of two successive leaves in a shoot apical meristem, and this results in a plant shoot being composed of repeated modules. Okay, so these modules actually consist of, first you have the internodes. Okay, so you have here the nodes, the different nodes. Okay, ito yung mga internodes na tinatawag. So these internodes do have cells that are produced by the meristem between successive leaf initiations. And then you also have the nodes. Usually it is attached with the leaf. And of course, the axillary bud. The axillary bud contains a meristem known as the lateral shoot meristem that forms at the base of the leaf. And of course, with its growth, the lateral buds immediately below the apical bud is suppressed, and the phenomenon is actually known as your apical dominance. So anything below it would actually, or the growth, there's always a suppression of the lateral bud growth development. Okay? So if you were listening to the videos that I have played a while ago, when you say lateral bud growth, those are the lateral outgrowths of a plant. So, yung mga nasa gilid like the leaves. And then when you say apical bud or the axillary bud, ito 'di ba yung ang growth niya is pataas. Okay? So, once a plant is growing pataas, of course, the lateral sides of the plant growth are usually suppressed. So, parang, kumbaga, nagkakaroon ng prioritization when it comes to the growth and development. So, mas priority ng isang plant yung growth niya pataas compared to the lateral sides of the plant. Okay? So, excuse me. So, this is actually an example of the apical dominance in plants. So, for example, if you cut a part of the lateral bud at the top, it would give rise to, or if you cut the apical part of a plant, ang magkakaroon naman ngayon ng growth is yung lateral sides. Okay? So, kumbaga, yung concentration of the concentration of the hormones for a plant to grow, nandun talaga siya sa apical shoot or the apical shoot meristem compared to the lateral sides. So, this is actually, an experiment that has been created to prove that that there is always an inhibition in the lateral outgrowths of a plant. So in here, pag pinutol mo yung kanilang apical na or the apical bud, and then you place an agar block. So instead of the plant growing upwards, what happens is that all of the hormones go down, and then the lateral outgrowths are being ano naman, sila naman yung nagkakaroon ng chance to grow laterally. Okay, so that's actually how the apical dominance is known in the growth of the plants. Okay, so the most likely affecting hormone in this type of growth is actually your auxin. Okay? So si auxin yung pinaka main na hormone natin na responsible for the apical growth of a plant. Okay? So the shoot apical meristems are actually small areas. They are rarely more than 250 micrometers in diameter and contain a few hundred relatively small undifferentiated cells that are capable of cell division. Cells actually leave the periphery of the meristem to form organs like the leaves or flowers, and are usually replaced from a small central zone of slowly dividing self-renewing stem cells of initials at the tip of the meristem. Okay? So again, most of the cell division that occurs with the growth of a plant, yung usual na fina-follow ng cells nila is cell elongation and enlargement. And we also have what you call the stem cells or what you call the initials at the tip of each meristem. So these initials are the cells that act as stem cells in plants. And promeristem is the central region of the meristem which contains these initials. And meristem cells are maintained in self-renewing state by cells underlying the central zone that form the organizing center. Okay? So, again, yung mga stem cells na tinatawag natin sa plants are what you call the initials, and they are usually found in the promeristems. And then these initials are actually do have what you call this organizing center. Of course, this center would eventually be developing to, like a pattern like part of a plant, no? Sila na yung magdedetermine kung anong type of cells yung mag-grow dun sa organizing center na yun. So again, we have here the central zone, which is colored in red. So this is the red portion of the organizing center. So this is from the Arabidopsis shoot meristem. So in this part, this is where usually the initial cells lie. Okay, so more likely the concentration of the stem cells are here. Okay, and then next we have here the peripheral zone. So this is the blue part. It contains the proliferating cells that will give rise to the leaves and the side shoots. And then we have here the rib zone, the one colored in yellow, which give rise to the central tissues of the plant stem. So with this rib zone, basically it would actually give rise to the central tissues of the plant, including the xylem and the phloem. Okay, so this is part of the organization center of an Arabidopsis shoot meristem. So that is how the cells are actually arranged. Okay? So the size of the stem cell area in the meristem is kept constant by a feedback loop to the organizing center. So numerous genes control the behavior of the cells in the meristem, and the first hormone or the first gene that we have here is the STM gene, or the shoot meristemless. Okay, so this is involved in specifying the shoot meristems in Arabidopsis, and its role is said to maintain the meristematic cells in an undifferentiated state throughout the adult meristems, but with its functional loss, it results in all meristem cells being incorporated into an organ primordia, in an organ primordia. So remember that a primordium starts an organ development in plants, right? So with this STM gene, usually it sets all um, these stem cells to just be in a general form. Okay? So if this STM gene do lose this capability of like controlling the changes in an undifferentiated cell, eventually this cells will be incorporated to become a primordia and eventually be forming organs from a plant or on a plant. Okay? So next we have the clavata gene. So the role is in regulating stem cell numbers, and mutation to these genes increase the size of the meristem as a result of an increase in the number of stem cells. So remember that normally the number of stem cells in a meristem is kept roughly the same throughout a plant's life by the division of the remaining stem cells. Okay, but with this clavata gene, okay, it actually increases the number of stem cells. Di ba nga sabi, ang plants, they do have this definite number of cells, and then from there, or from those number of stem cells, nag-divide na lang sila to be able to form the different organs. So, wala nang nadadagdag or wala nang nababawas. Okay? But with the clavata, it actually increases the number of stem cells in a plant. Okay? So next, we have here what you call your wushel. So this is an expressed homeobox transcription factor by the Arabidopsis organizing center, which actually is required to produce signals that give overlying cells their stem cell identity. So mutations will result to termination of the shoot meristem and cessation of growth as a result of the loss of stem cells, while its overexpression increases the stem cell numbers. Okay? So this is like the homeobox gene also of or in animals. Okay? So they give out, or this Wushel gene produces signals, okay, for their stem cell identity. So next, you also have the clavata 3. So this is also expressed by the stem cells, and it encodes a secreted protein that acts indirectly to repress your wushel. Okay? So basically, these are the components in the regulation of the stem cell population in your meristems. Okay? So the fate of cells from different meristem layers can be changed by changing their positions. So as well as being organized into central and peripheral zones, the apical meristems of a dicotyledon such as the Arabidopsis is composed of three distinct cell layers. So the first one is the L1, which is the outermost layer. It is around 1 cell thick. You also have the L2. L2 just beneath L1, it is also just 1 cell thick. And both of them actually, or the cells within these two layers actually divide by anticlinal divisions and are often known as the tunica. Okay? And then for the L3, it is the innermost layer. This is actually where the cells divide in any plane and make up the corpus. Okay, so this red or the peach colored one is the L1, L2 is the yellow one, and then the L3 is the green one is the corpus. Okay? So L1 and L2 makes up the tunica through anticlinal divisions. Ito yung tinatawag natin na, if you remember, the growth of the cells are actually perpendicular to the layer. And then you also have, again, the L3, which actually divides in any, any direction of the plane and makes up the corpus of a plant. Okay? So, cell fate has been determined by generating chimeric tissues. So, chimeras are composed of cells of different genotypes and are made by treatment with radiation or chemicals like colchicine. So, we have periclinal chimeras that have one of the three layers marked indifferently. And basically, the layer has different cell type but compared to the other layers of the meristem. So with periclinal chimeras, mapapansin nyo na lang na yung type of cells ng isang layer ay different from the other two layers. Okay? So in angiosperms, L1 becomes the epidermis while L2 and L3 produce cortex and the vascular tissue. L2 also produces the leaves and flowers, and L3 contributes mainly to the stem. Occasionally, L1 or L2 cells divide periclinally that invade a new layer and adopt the fate of the new layer, which is very regulative. Okay? And the next, apart from the periclinal chimeras, we also have what you call the mericlinal chimeras, which are the result of X-irradiation or mobilization of transposons. So this transposon, these are actually the jumping genes that are located in the genome, and they can actually move from one location of the genome to another. Okay? So with that, the entire sector of a plant is marked by a clone. So, kumbaga itong transposon na to, para siyang nagmo-move or nagmo-move talaga siya from, yung characteristic na meron siya, i-move niya sa ibang part ng genome. Kaya nagkakaroon ng clones. Okay? So, these have been used to produce probabilistic fate map in maize and Arabidopsis. And when you say probabilistic fate map, this is actually a model where fate probabilities of progenitor cells differentiating into different terminal states can be predicted. So, parang napakita ko na sa inyo before what is a fate map, 'di ba, yung may mga boxes and colors. So, ganun din sa plants usually. Okay? So, this figure that you see here is a periclinal meristem chimera composed of cells of two different genotypes. So, sabi nga, when you say that there is a periclinal meristem, the types of the cells of the different layers are different from each other. So in here, you see that L1 has 2 N nuclei, and then the second layer has 4 N nuclei. So with this one, what happens usually, for example, what we have here is, of course, a plant that has different colors on the leaves. Okay, so this is this is also a manifestation that there is a different phenotypic expression because of these transposons or because of these jumping genes. So instead of the plant only having one color, nagkakaroon ngayon ng variations. Okay? So next, this figure shows you the clonal analysis of the maize embryo, where it can be divided into 6 vertically stacked domains, and each containing a set of initials that can give rise to a particular part of the plant. So this is the probabilistic fate map of a maize embryo. So all you have to remember when it comes to the probabilistic fate map is that, of course, in this part of the section of the embryonic shoot meristem, it is divided into 6 domains. So this is pertaining to the different colors that you see here. So this part, or the level of each domain, comparing or correlating it to the section of the embryonic shoot meristem, it would actually give rise to a specific internode. Okay? So for example, we have here domain 1, which would give rise to internode 6, 7, 8, and 9. And then on an adult plant, it would give you the lower parts of the um, growing maize plant. So, in domain 6, it would actually give rise to the central spike or the tassel base. So, ito yung pinaka-tip talaga ng mature maize plant. Okay? So, for the female inflorescence, usually it would be seen somewhere in between internode 12 to 15. So, that is the domain 3. Okay? So, madali naman siyang intindihin, no? Madali namang intindihin yung probabilistic fate map because of the different colors and what would be the resulting adult organs in a plant. Okay? So, apart from the maize plant, we also have here a probabilistic fate map of the embryonic shoot meristem of Arabidopsis. So coming from this layer of the meristem, you know, the different colors will also give you the idea on how these cells would actually travel in the during the growth of the meristem, and of course what would be the equivalent organs in a growing or in a mature Arabidopsis plant. So for example, this cells that are colored in peach and labeled as number 7. So saang part sila ngayon nag-grow ng Arabidopsis plant? Okay? So majority of the cells, the colored or the cells that you see here on the layer of the meristem are actually on the basal part. So that includes the cells with number 1, 4, 2, 6, 3, and 5 numbers. Okay? And then the rest are already found on the higher parts of the Arabidopsis plant. Okay? So um, that's how you map, no, the fate of the different cells in a growing meristem. Okay? So next, meristem development is dependent on signals from other parts of the plant. So as we saw earlier, the apical meristem of the maize give rise to a succession of 60 of nodes, 16 to 22, and terminates in the male flower or the tassel. So isolated meristems do not retain memory of how many nodes are produced and will generate a complete set of nodes. So the number of the nodes is actually determined by the interaction of the meristem with the plant. Therefore, development is not determined in the embryo with respect to the number of nodes it will form, but will also involve signals from the rest of the plant of the meristem. Okay? So, di ba nga guys, hindi naman lahat kasi ng plants do have the same number of nodes in it when they grow. So, wala talaga siyang pattern unlike in animals. So, kaya nagkakaroon ng variation sa growth ng plants. More likely, they are dependent on the signals, okay, from the different parts of a plant. So, gene activity patterns, the proximodistal and adaxial-abaxial axis of leaves developing from the shoot meristem. So, the first indication of leaf initiation is the meristem is usually a swelling of a region to the side of the apex which forms the leaf primordium. So this is the result of an increased localized cell multiplication and altered patterns of cell division, and also reflects changes in polarized cell expansion. So the axes that relate to the future leaf is actually the following. First, you have the proximodistal axis, which indicates the base of the leaf to the tip, and then the adaxial abaxial, which is the upper surface to lower surface. Usually, we know it also as the dorsal and the ventral sides of a future leaf. Okay? So, examples of the Arabidopsis abaxial gene is the filamentous flower or the fill. And then for the adaxial, we have the fabulosa or fab. We also have the fabuluta or fav. And then the revoluta or the rev. So those are just the examples of genes that are responsible when it comes to the genetic activity of and patterning in the developing leaf. Mhm. So the regular arrangement of leaves on a stem is generated by a regulated auxin transport. So as the shoot grows, leaves are generated within the meristem at regular intervals and with a particular spacing. So phyllotaxy or phyllotaxis is actually the term used for the leaf arrangement in a plant. So leaves can occur singly at each node. It can also be in pairs or in whorls, in three or more groups or kumbaga in three or more na clusters or clumps. Okay. So this also involves lateral inhibition and often produces a helical pattern of leaves on a stalk. So the figure 14.23, the auxin-dependent mechanism of phyllotaxis in Arabidopsis. So basically, the parts wherein the colors are brighter in green, this denotes that there is a higher auxin concentration. The ones that, or the part wherein it is colored light green, there is a low concentration of auxin. And then of course the arrows would give you the idea um, of the direction, okay, of how this auxin um, actually mechanizes inside a or in the developing in the developing plant. Okay, so auxin is transported through pin proteins towards areas of high auxin concentration at which an organ primordium will form. So cells around the developing primordium become depleted of auxin, which causes the polarity of the pin proteins to reverse so that auxin then flows away from the primordium. So the red arrows indicate the direction of the pin polarity. It is proposed that this pattern of auxin circulation could set up the or, or could set up the regular pattern of leaf and flower formation from the meristems. Okay, so this actually works, or this auxin actually works with your pin proteins. Okay. So root tissues are produced from Arabidopsis root apical meristems by a highly stereotyped pattern of cell divisions. So root meristems resemble shoot meristems but have two important differences. The first one is the root cap that covers the root meristem for protection. And second is that no segmental arrangement is seen with the node internode node module. Okay? So the root is set up early in the late heart stage embryo by a set of initial cells, and each column of the root cells originate with a specific cell in the meristem via a specific pattern of cell division. Nevertheless, this process is under regulatory control as laser ablation of developing root cells result in normal tissue. At the center of the root meristem is a quiescent center of cells that do not divide, and there is no obvious segmental arrangement of root as seen in the node internode leaf module of a shoot. Okay, so this is actually now the difference of the the developing meristems. Okay? So, kung sa shoot meristem, meron tayong mga modules na tinatawag. Sa root naman, wala siyang ganun. Okay? More likely, controlled lang yung development niya and walang nangyayari na segmental arrangement compared to the shoot meristem. Okay? So this one shows you now the different structure of the root tip in Arabidopsis. So figure 14.24 is the structure of the root tip in an Arabidopsis. So roots have a radial organization. And in the center of the growing root tip is the future vascular tissue, including your protoxylem and protofloem. So, dito yung vascular tissue. So, this is surrounded by further tissue layers that include the epidermis and, of course, the root cap. Okay? So, another figure is the fate map of the root regions in the heart stage of the Arabidopsis embryo. So the root grows by the division of a set of initial cells. So coming from this heart-shaped embryo, the root meristem comes from a small number of cells in the heart-shaped embryo, where each tissue in the root is derived from the division of a particular initial cell, and at the center of the meristem is again here, you see also the lateral outgrowths of the root hair. You also have here the root cap. Okay, so dyan nag-uumpisa yung ibang outgrowths. Okay, so coming from, if you also make use of the fate map, okay, so the different colors would actually give rise to the particular organs of the plant as you see here. Okay. So for the summary of the meristems that give rise to all adult tissues in shoot meristems, of course, there's always cell fate is actually determined by its position. And then shoot meristem give rise to stem internodes, leaves, and flowers. And arrangement of the leaves on stem may be due to an auxin-based mechanism at the shoot apex. While for the root meristem, it is a stereotype division of initials along different planes, and the root meristem give rise to all root structures. Okay? So this is actually the last slide for our discussion for meristem. So let's go directly now to the flower development. So flowers contain the reproductive cells of higher plants and develop from shoot meristem. So in most plants, the transition from a vegetative shoot meristem to a floral meristem produces a flower is largely under environmental controls. Examples is actually your day length and temperature being important determining factors. Okay? So complex mechanisms are involved to pattern the flower to form the sepals, petals, stamens, and your carpels. So in here, we are already, we will be determining what are the factors that would actually affect the flower development. So there are three classes of genes that are involved in basic flower patterning. So the first one are the organ genes, which identify different floral organs, which are much like homeotic selector genes in animals. So if you compare it to animals, that this is their, type of genes naman sa plants. Okay? And then next, we have the cadastral genes, which set boundaries of organ gene expression. And then next, we have the meristem identity genes, which convert shoot meristem into floral meristem. So examples of key regulatory genes of floral induction for Arabidopsis are the leafy genes. This is a meristem identity gene, and another related gene is the floricola for antirinum. Okay, so I would just like to add an information about the leafy and floricola genes because I have used them before in doing a study about familial a family of plants, specifically the family or the group of the what they call this the plant group of basta it's a plant that I've studied during my MS, no. They actually utilize this leafy gene and floricola gene to to trace the lineage of this particular plant. And apparently these genes are very short, no, very short lang in sequences, pero they give out so much information about the characteristic of a certain plant. 'Di ba nga sabi natin if we compare different species of plants or even in animals, ang pinakamarami talagang information na pwedeng nating i-compare is already within their DNA sequences compared to the physical attributes that you see on a plant. So, itong floricola and leafy gene na to, usually ginagamit din siya to compare different plants, especially to plants that do have, alam niyo yun, yung parang napakadami nilang variations. Pero, they fall into a specific characteristic of the gene, kaya natatawag silang member nung group na yun or nung family na yun. Okay? So, ginagamit siya as a molecular marker as well to identify other maybe plants that could be part of that certain group. Okay? So next, homeotic genes control organ identity in the flower. So shoot meristem converts to inflorescence meristem, which can form one or more floral meristems. So the individual parts of a flower each develop from a floral organ primordium produced by the floral meristem by cell differentiation and enlargement. So again, you have the floral organ primordia, which will give rise to the floral meristem. Okay? So there are four concentric walls that reflect the order within a floral meristem. The first one is the sepals or the whorl one. This arise from the outermost ring of the meristem. And then the second one would be the petals or whorl 2 from the next ring. And then stamens for whorl 3, which includes which will give rise to the male reproductive organ coming from the inner ring. And then the carpels or the whorl 4, this is the female reproductive organ coming from the center. Okay? So in Arabidopsis, there are actually 16 separate primordia, giving rise to a flower with 4 sepals, 4 petals, 6 stamens, and a pistil made up of 2 carpels. So figure 14.28 is a representation of the arrangement of the parts of the flower, showing the number of flower parts in each whorl and their arrangement relative to each other. Okay, so this one. So these are the concentric whorls that would actually give rise to the floral meristem. Okay? So mutations can also happen in flowers in their floral identity genes, causing homeotic mutations in which one type of flower part is replaced by another. So this identify flower organ identity genes and help determine their mode of action. So we have here, of course, the homeotic mutats. Okay. So there are 3 classes of homeotic floral mutations, and each affect the organs of 2 adjacent whorls. So the the first one would be the class 1, which includes apatella 2 and affects whorls 1 and 2 in such that sepals are replaced by carpels and petals by stamens. And then class 2, such as in pistillata and apatella 3, it affects the whorls 2 and 3 and have petals replaced by sepals and stamens by carpels. And then class 3, we have the agamus that affects the whorls 3 and 4 and have the reproductive organs replaced by sepals and petals. Okay? So hindi lang sa animals nagkakaroon ng mutations, it's also possible for plants to have certain mutations. Okay? So, the model of gene activity in the floral organ development is or do have the floral meristem divided into three overlapping regions A, B, and C. Okay? So, region A contains whorls 1 and 2, B contains whorls 2 and 3, and C covers whorls 3 and 4. So this combination of A, B, and C regulatory functions give each whorl a unique identity. And in addition, the A and C regulatory functions must be mutually exclusive such that it actually prevents C activity and vice versa. So the floral meristem region with A function only produces sepals, A and B functions produce petals, B and C functions produce stamens, and C function only produces carpels. So homeotic mutants eliminate either A, B, or C functions. Okay? So apart from having, the homeotic mutant classes, we also have this organ development regions. Okay? So, a model of gene action controlling flower patterning is consistent with the combinatorial organ identity specification. So, again, in class 1, A is disrupted and C is expressed in whorls. So, in return, it will develop carpels, stamen carpel pattern. Okay, sorry for the typo error. So that will be stamen. Okay? So for example, it is expressed in apetella 2 mutation. So class 2, only A function in whorls 1 and 2 and C in whorls 3 and 4. So what would transpire would be a plant or a flower having a sepal, carpel characteristics. So these are usually found in the pistillata and appellata 3 mutations. And then class 3, C is disrupted and A is expressed in all the whorls. So in return, it would result to a sepal, petal, sepal type of flower. So it is found in an agamus type of mutation. So the combination of functions modify the ground state to cause flower development. And the agamus protein is expressed in the central whorls, while apetala 3 is expressed in the outer whorls. So these are the images that would actually explain to you how these mutations happen. So in figure 14.29 here, this is the homeotic floral mutations in Arabidopsis. It shows the homeotic transformation of whorls which are presented in set and can be compared to the wild type arrangement. So in here, this is the Apetala 2 mutation is to see it doesn't have really the petals itself. For Apetala 3 mutation, no, wala siyang mga yung ibang parts niya wala. And then this is the agamus mutant. Okay? So the figure below shows the three overlapping regions of the Arabidopsis floral meristem that have been identified by the homeotic floral identity mutations. So region A corresponds to whorls 1 and 2, B again corresponds to whorls 2 and 3, and C to whorls 3 and 4. So this actually gives you the idea that these regions do actually overlap with itself. Okay? So just follow also the colors of the schematic diagram for you to to visualize properly how these mutations happen. So, this is already, or this is the wild type. So, ibig sabihin ng wild type, ito yung dapat or the original type of a flower of an Arabidopsis. So, dapat ganito yung kanyang arrangement. Okay? But then, what happens in the different mutation is like this. So, for example, we have the Apetala 2 mutant. So, this is actually how the expression of the homeotic floral mutations do look like. So, wala siyang B, no? Wala siyang A. Okay? So, kung wala siyang A regulatory gene, so wala siyang mga sepals. Okay? Katulad nito, ito, Apetala 2 mutant, wala siyang mga sepals. And then, we also have the Agamus mutant. Ito naman, wala siya ng mga gitna, mga stamens or the reproductive organs. So, what happens to an Agamus? It looks like this. More on the, ano lang talaga siya, the petals and, of course, the sepals, pero wala siyang reproductive organs. Okay? So, the figure to the right shows the same regions and their activities during the different mutations compared to the wild type. Okay, so this is just a bigger representation of how the mutation happens in Arabidopsis. So if this is the wild type, this is what happens with the Apetala 2, this is the Agamis mutant, this is the Apetala 3 or Pistillata mutant, and then this is the Apetala 2 or Apetala 3 mutant. Okay, so those are the probabilities of mutation in an Arabidopsis plant. So the floral identity genes encode homeotic proteins. So Apetala 1 and Agamus share a conserved DNA binding domain, the MADS box. So the MADS box proteins act as transcription factors and are present in plants, fungi, and animals. This act as a homeotic selector gene in a manner similar to the Hox gene in animals. And in addition, the curly leaf gene in Arabidopsis is like the polycomb family of genes in Drosophila. So these curly leaf proteins act to cause the stable repression of the homeotic genes in plants. And we also have here the Superman. This is a cadastral gene that acts to maintain the boundary between the third and fourth whorls. Okay, so those are just the type of genes that encode for homeotic proteins. So the transition of a shoot meristem to a floral meristem is under environmental and genetic control. So flowering plants first grow vegetatively, during which time the apical meristem generate leaves, then triggered by environmental signals. It can be externally or internally, and examples would be the increase in day length or switching to reproductive phase, and so on and so forth. So there are so much factors that could actually affect this transition. Okay, it can be either externally or internally. So there are two types of transition from vegetative growth to flowering. The first one is the bideterminate transition, wherein the inflorescence meristem becomes the terminal flower. And the second one would be the indeterminate transition, wherein the inflorescence meristem give rise to a number of floral meristems, as in Arabidopsis. So, leafy and Apetala 1 are floral meristem identity genes that are expressed in response to floral inductive signals. And mutations in floral meristem identity genes partly transform flowers into shoots. Okay? So, ayun. Multiple talaga, or yung floral meristems of Arabidopsis are um, are multiple. Okay? Are multiple compared to other types of plants. So the transition is also affected by what you call the short and long days. Okay? So in Arabidopsis, flowering is promoted by increasing day length, which predicts the end of the winter and onset of the spring and summer, and this behavior is called the photoperiodism. So there are actually some plants that they are more likely dependent um, on the day length. 'Di ba kapag ka nagbabago kasi yung day length natin guys, 'di ba? It also gives us the idea that nagbabago na ng season. Okay? So ganun din sa plants. So they call it the photoperiodism. And then next, we also have the vernalization. This is the phenomenon being applied to other strains where flowering is promoted by when a plant has been exposed to long period of cold temperature. So with vernalization, more likely it's being affected with the temperature in the environment. Okay? So plants also do possess the circadian clock. This is the internal 24-hour timer that causes many metabolic and physiological processes in plants. For example, the inclusion of the expression of genes. So we have here what you call the constans. This is one of the genes being regulated by the circadian clock, which is the key gene in controlling the onset of flowering and provides the link between the plant's day length sensing mechanism and production of flowering signals. So, usually, the peak of the constans expression or the CO occurs towards the end of the afternoon, meaning in longer days, peak expression occurs in the light, whereas in short days, it will already be dark at this time. So in the dark, CO proteins is degraded and so the circadian control makes sure that the CO only accumulates to high enough levels to trigger the flowering pathway when light conditions are favorable. Okay, so more likely, proteins are degraded in short days and not during the long day periods. Okay, so constants or the CO gene is also activated by the gene known as the flowering locus T or FT that acts as a flowering signal. Okay. So most flowering plants are hermaphrodites but some produce unisexual flowers. Okay? So unlike animals, plants do not set aside a germ cell in the embryos and that are only specified when a flower develops. In meristems, it can give rise to a germ cell of either sex and there are no sex chromosomes. So great majority of flowering plants give rise to flowers that contain both male and female sexual organs in which meiosis occurs. So most of the flowering plants are hermaphrodites, bearing flowers with functional male and sexual organs only. There are actually only about 10% of the flowering species that do have a single sex organ. Okay? So as in Arabidopsis, they actually undergo the so-called double fertilization. So it involves two sperm cells. One fertilizes the egg cell to form the zygote, while the other fuses with two polar nuclei that form the endosperm. So sex determination becomes visible when the flower is still small, with the stamen primordia being larger in males and the pistil longer in females. So usually, with this outgrowths, the smaller organs do usually degenerate. So kung ano yung mas malaking organ pagka nagde-develop sila, yun na yung magde-determine kung ano yung sex type nila. Okay? So gibberellic acid is actually a plant hormone which its concentration is said to be responsible for sex determination in plants. So for the summary of the flower development in Arabidopsis, again, we have the vegetative meristem, and then there should be environmental signals to be able to form the inflorescence meristem. And then to form the floral meristems, there should be the presence of the meristem identity genes. And coming from the floral meristems, there should be a concentric, there is always this identification of the three concentric overlapping regions of the gene expression that's established. And flowering consisting of four concentric whorls like the sepals, petals, stamens, and the carpels. Okay. So for more information, or if you would like to read the certain chapter of plant development, the reference that we have used for this lecture is the Wolpert et al. Principles of Development, 5th edition by Oxford University Press. So if you would like to read more about the plant development, you may want to read or grab a copy if you can, for you to read more of these lesson that we have for plant development. So that actually ends the lecture for plant development. If you have any questions, just let me know and I'll be answering it. Okay? Thank you.